Research suggests there is an unusual form of cell death causes severe COVID-19 lung damage. Unusual form of cell death causes severe COVID-19 lung damage. This is by Megan Redshaw, JD, via the Epoch Times. SARS-CoV-2 infection may cause severe pulmonary conditions, as we know, such as pneumonia, inflammation, and acute respiratory distress syndrome. But until now, researchers have not understood the driving force behind lung damage caused by COVID-19. In a recent published paper in Nature Communications, researchers at Columbia University discovered that a relatively new form of iron-dependent cell death called ferroptosis is the underlying mechanism causing extreme and potentially fatal pulmonary conditions in COVID-19 patients. This finding adds crucial insight to our understanding of how COVID-19 affects the body that will significantly improve our ability to fight life-threatening cases of the disease. Brent Stockwell, chair of the Department of Biological Sciences at Columbia and co-lead author of the study, said in a new release, "What is ferroptosis? Human cells have powerful mechanisms for maintaining survival, but when those mechanisms become impaired, lipid peroxides generated through normal metabolic activities can accumulate to toxic levels, damage cell membranes, and kill cells throughout the ferroptosis." Ferroptosis was first identified in 2012 by Mr. Stockwell and differs from apoptosis, the most common form of cell death that occurs with aging and in some disease processes. Ferroptosis is an unusual type of cell death linked to altered iron metabolism, glutathione depletion, glutathione peroxidase 4 inactivation, and increased oxidative stress. Since proposing the concept, Mr. Stockwell's lab has demonstrated that ferroptosis can occur in healthy contexts as part of the body's normal processes. For example, cell death can remove cells infected with SARS-CoV-2, inhibiting the replication and spread of the virus, or counteract diseases like cancer, which involves rapid cell growth. On the other hand, ferroptosis can be destructive, attacking healthy cells in patients with neurodegenerative diseases or causing severe manifestations of COVID-19, possibly even long COVID. The ability to stop ferroptosis could pave the way for treatments that improve COVID-19 outcomes, the paper's findings suggest. The study? Researchers obtained lung tissue samples from the autopsies of patients who died of respiratory failure caused by severe COVID-19, including those with and without acute lung injury, ALI. Samples were also collected from patients who had mild COVID-19 and recovered. Control samples were obtained from pre-pandemic healthy patients who showed no signs of SARS-CoV-2 infection or lung damage. Samples from autopsies with ALI and without a history of SARS-CoV-2 infection were also analyzed. Additionally, serum ferritin records were obtained, as serum ferritin is elevated in critically ill COVID-19 patients, correlates with disease severity, and supplies the iron that drives ferroptosis. Ferritin is a protein found inside the cells that stores iron and allows the body to use it when needed. The researchers analyzed cell death markers on COVID-19 lung tissue samples obtained from the autopsies and found distinct molecular features of ferroptosis in severe lung pathologies. Moreover, samples revealed elevated ferroptosis markers, including iron dysregulation, lipid peroxidation, elevated phospholipids, and depletion in polyunsaturated fatty acid tails, PUFAs. PUFAs, or polyunsaturated fatty acids, while essential for cell membranes and immune regulation, are highly unstable and inflammatory. Due to their sensitivity to oxygen, PUFAs can be oxidized by free radicals and broken down into toxic derivatives. In a secondary analysis, Syrian hamsters were initially inoculated with the same strain of SARS-CoV-2 found in patient samples. Researchers analyzed lung samples and found that ferroptosis occurred at an early stage of lung disease and accompanied disease progression. To confirm their findings were caused by ferroptosis, the researchers measured cell death markers associated with other forms of cell death. They did not detect an increase in these markers in any lung samples, indicating that ferroptosis was the major cell death mechanism detected in fatal COVID-19 lung samples. Altogether, these data suggest a strong correlation between ferroptosis and COVID-19 lung pathology, and ferroptosis inhibition may serve as adjuvanted therapy to reduce lung injury, the authors wrote. A May 2023 paper published in Frontiers supports the paper's findings. In their review, researchers found that dysregulated cell death from ferroptosis can lead to uncontrolled cellular damage beyond the lungs and plays a role in the multi-organ complications observed in COVID-19 patients with severe disease. And this is by Megan Redshaw, JD via the Epoch Times on Zero Hedge. Please leave your comments. Thank you for your support. Finally, support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.